welcome to Virtual Food Safety Office Hours. I'm Abby Snyder. I'm an Associate Professor of Microbial Food Safety in the Department of Food Science here at Cornell. And today we are discussing updates on highly pathogenic avian influenza virus. So this is, I think, the fourth or the fifth session kind of in this series on this topic. The previous recordings of the presentations are available through the IFS YouTube channel. And then joining us today for this newest installation in the series, we have Dr. Diego Dial, who's an associate professor in population medicine and diagnostic sciences. His lab investigates the mechanisms underlying virus-host interactions and applies that knowledge to enhance control of infectious disease in domestic animal species. So he'll be one of the initial presenters we have today. Also joining us, we have Renata Ivanek, who's a professor also in population medicine and diagnostic sciences. Her research applies data and model-driven approaches to controlling infectious disease and optimizing food production systems. And presenting, we have Catherine Coble. So she is a PhD student in epidemiology advised by Professor Ivanek. So after Diego, we'll have a presentation from Catherine on quantitative risk assessments. And then following the presentations, we'll be having a discussion panel. Joining us on that panel, in addition to the speakers, we have Dr. Nicole Martin, a research assistant professor in the Department of Food Science. She is also the associate director of the Milk Quality Improvement Program. We have joining us again, Dr. Alicia Fry, who's an associate professor within the Department of Population Medicine and Diagnostic Sciences and with the Animal Diagnostic Center. And then finally, we have Dr. Rob Lynch, DVM, who is a dairy herd health management specialist with ProDairy at Cornell. So with that, I'm going to turn it over to Diego, who's going to kick this off with a few updates on current numbers and new variants.
Hey, thank you. Thank you, Abby, and thanks everyone for being here today. I'm going to try to share my slides here. Let's see. Can you guys see my slides?
Yes, they are not in presenter mode quite. There you go. Perfect. Thank you.
Okay, perfect. So I put a few slides together here just to give an overview of where we are with this situation. As everybody know, by now, I mean, the spillover of this virus into dairy cattle was first detected in March of 2024 with the data at the time, clinical data and epidemiological data suggesting that most likely that spillover had happened, anywhere between January and February of 2024. The initial spillover was detected in Texas, and initial clinical cases in affected animals was detected in Texas. And following that detection in Texas, the virus spread to multiple states, most likely, with the data pointing to animal movement being one of the strongest links to movement of the virus throughout the US. This is the current situation. I just downloaded this map from the USDA website, a few hours ago. So the initial outbreak initiated in Texas, and then was the subsequent outbreaks were detected in New Mexico, Kansas, Michigan, Ohio, and North Carolina within a month time frame of the initial detection. But as you can see now, we have over 970 confirmed affected herds in 17 states. So the virus basically spread to multiple states and it has been detected and officially confirmed in at least 978 states. I personally believe that the number of affected herds is much higher than this. And the reason why we have only 978 confirmed cases is the fact that there's not a whole lot of testing that is happening to detect this virus in dairy cattle. If we look at the cases in the last 30 days, so this picture actually changed also recently. If we had looked at this map 30 days ago, we would see that only California was affected at the time. But very recently, the USDA, through a wide surveillance program that they implemented by testing milk silos, they actually detected the presence of the virus in Nevada and Arizona, and also in Idaho. So these are the 4 states in which cases or new cases have been detected in the last 30 days of the outbreak. One of the interesting findings about the Nevada and the Arizona cases or herds that are affected is that the virus that was detected in Nevada initially and then subsequently in Arizona, it's a virus that belongs to a genotype that is different from the original virus B313 that was detected in Texas and that until January of this year was the only virus or genotype that had been detected in dairy cattle. The new virus or the new genotype that was detected in Nevada and Arizona belonged to the genotype D1.1. And I know that this is probably very complicated for a lot of the audience here, but the genotypes of influenza are based on the basically the makeup of the genome of the virus. This is a virus that has a segmented RNA genome, so it's made up of 8 segments that encodes obviously the proteins that the virus needs to replicate in the host. And because of the segmented nature of the virus and the fact that certain species can be infected by more than one virus, usually when that co-infection happens, those genome segments from 2 different viruses can be shuffled as the virus is replicating in that host species, and that usually originates a completely new virus that then is classified as a new genotype based on the genome segments that new virus has acquired. So why is this important, and why am I highlighting that? This discovery of the genotype D1.1 in Nevada and Arizona also provide evidence of 2 independent spillovers of the virus into dairy cattle. So it's not the same virus that spilled over in Texas and then spread to the other states and that is infecting cows in California. These are 2 independent events in which the virus spilled over from wild birds, most likely, into dairy cattle. So we now have documentation of at least 3 events that in which the virus jumped from wild birds into cows. And so this is the official report that came out on January or early February, describing the detection of the D1.1 genotype by USDA and APHIS. So this is just a quick cartoon showing the reassortment that happens that originates the different genotypes of influenza. As I mentioned, these are 2 different influenza viruses. When they infect the same host cell, the genome segments are released in the cytoplasm as the virus is replicating, and those can shuffle and originate viruses with a different genetic makeup. And that's how this D1.1 genotype originated. This is very common in influenza, especially in wild bird populations where we have multiple different viral strains co-circulating. So this process of reassortment and emergence of different viruses is very common. And then this is data showing some of the data that we have here that we collected here at the HTC using sequencing, where we do genomic surveillance to monitor what are the genotypes that are circulating in the samples that test positive here. Most of these samples are samples that from wild birds that were tested at the HTC. As you can see here, this genotype D1.1 that was detected in dairy cattle in Nevada and Arizona is now the dominant genotype that we are detecting here in New York and in the Northeast states as well. This virus is the dominant genotype of influenza now circulating in the wild birds, which are the reservoirs for the virus. So why is this? What is the public health impact of this? We all know that this is a zoonotic virus. Up until early this year, we had 69 or 68 cases of confirmed human cases of influenza infection. And in January, I think everybody remembers that there was one case of a person in Louisiana that actually ended up dying of influenza infection. And the virus that was detected in that patient is the genotype D1.1. So this genotype seems to have a higher virulence to humans. To date, there's no evidence for human-to-human transmission. There's another case that was detected, reported a little earlier than the case in Louisiana in a child in Canada that also presented with more severe clinical presentation when compared to the D3.13 infections that have been described in the US, where most people presented mild clinical signs, mild respiratory signs, or conjunctivitis. So it is, the circulation now of this genotype in dairy cattle, it's concerning because it seems like this virus has, higher virulence properties in humans, and also the fact that the virus is the dominant genotype circulating in wildlife, in wild birds especially, also raises concerns of potential for additional spillover events in states where we haven't yet detected the virus, including in New York. We have not detected H5N1 infections in dairy cattle, but the virus is circulating in the wild bird population. I think with that, I'm gonna stop here. I just wanted to give a brief overview of where we are currently on this situation. I'll pass it on to Kat for her presentation.
Thanks, Diego. Over to you, Catherine.
Thank you. All right, can everyone see my slides okay?
Looks good.
Excellent. All right. Hello, everyone. My name is Dr. Catherine Cable. I am a dairy veterinarian and a PhD student in the lab of Dr. Renata Ivanek up in the vet school. We are an epidemiology and food safety lab, and we're collaborating with this amazing team here to use computer modeling tools powered by real-world data to investigate the risk of H5N1 infection in people consuming fluid milk, raw or pasteurized. We've heard lots of great information today, so I won't go into too much detail on the background because we all know why we're here. H5N1 has jumped species from birds into dairy cattle, and it's continuing to spread amongst herds. And we know that these cows shed the virus heavily in milk. And while there have been no reports of people becoming sick from consuming contaminated milk, we do know that the strain of H5N1 is infectious in people. And there are both laboratory studies and anecdotal evidence that raw milk is infectious in multiple species, including cats, mice, and research monkeys as well. Which leads to our two research questions. What is the risk of H5N1 infection from drinking raw or pasteurized fluid milk? And what food safety interventions can mitigate this risk? We developed two risk assessment models, so computer programs, in a software called At Risk. One model represents the US pasteurized fluid milk supply chain, and the other represents the raw fluid milk supply chain. And we modeled these separately because the cold chains and the overall processing are fundamentally different. But both models have the same 3 steps: herd dynamics, processing and packaging, and consumption and infection. In herd dynamics, the model will simulate milk production and herd sizes, including how many sick and healthy cows there are. Once the milk is mathematically generated, it goes on to whatever processing or packaging there is. In the pasteurized milk model, the amount of virus in the bulk tank here is reduced 12 log per the USDA estimate of high temp short time pasteurization. Milk goes into gallon packages and then it's then distributed to consumers. In the raw milk model, we have 2 different purchasing pathways: buying raw milk at retail, which is legal in some states, and buying raw milk directly from the farm, which is legal in some other states. For dose response, so the equation that determines how likely someone is to get sick based on the quantity of virus in their 8-ounce serving, not much is known, right? This is an unprecedented situation. So we use our best scientific judgment in developing that aspect of the model. And this is a shorter presentation, so in the interest of time, I will mention that that parameter is an area of active investigation in this project, and it's discussed further in the paper. I mentioned that one of our research questions was investigating how different interventions can change risk. We do that with a method called scenario analysis. In the pasteurized milk model, that intervention is, of course, pasteurization. In the raw milk model, we examine daily PCR testing of herd bulk tanks alone and in combination with better pre-harvest identification and diversion of sick cows. And we define the diversion of sick cows as recognizing that a cow is unhealthy and unfit to contribute to the milk supply before her milk hits the bulk tank. Here are our projections for risk in our baseline scenario. I'll briefly explain how to interpret these so-called violin plots. First, please do note that this is a log scale and that it is inversed. So lower numbers are better in that they mean less risk. And where the violin kind of bunches up is where values are concentrated, and a long tail that looks like a stingray means that there's kind of a long train of values. So based on our current model, the risk of H5N1 infection from consumption of pasteurized milk is extremely low. It's about 1 in 176 trillion servings. Raw milk is noticeably higher, but it's also quite spread out, right? The median is about 1 in 885, but it can be as low as 1 in 142 billion. Finally, there really is not much difference between the 2 purchasing pathways. And a different way of looking at it, in our model, an iteration represents a full calendar year of pasteurized fluid milk production and consumption. So in our simulations, 99.5% of iterations predict 0 H5N1 infections in people consuming pasteurized milk, and the remaining 0.5% of iterations predict 1 to 3 cases per year. In our baseline scenario, I mentioned that we use that 12 log reduction, but we wanted to manipulate that because it is kind of hard to demonstrate 12 log reduction if your initial inocula are like below a 12 log concentration. So our wonderful collaborators were performing some heat treatment assays, and 6 log was about the most concentrated inoculum they had, which did fully inactivate with HTST for the record. Then we ran simulations beginning with a 6 log reduction and going up in multiples of 2. And this demonstrates the intimate relationship between pasteurization efficacy and the risk of infection from pasteurized fluid milk. And this means that if we are systematically overestimating our log reduction, or if a pasteurization failure goes undetected, risk could be higher, on the order of 1 in 2.7 million. Here's our other scenario analysis. This one is for raw milk, and this is the farm store purchasing pathway. Although again, the two pathways were very, very similar in terms of risk. Here we investigated the percent diversion of sick cows and also the PCR testing of farm bulk tanks. So these three violins are improving from a 25% chance of diversion to 50% and to 75%. And while numerically there is a slight difference in risk, it's kind of hard to appreciate with this plot. It's not nearly as impactful as jumping from no PCR testing to yes PCR testing. These findings emphasize the importance of pasteurization during the emergence of a novel milk-borne pathogen. It's also identified or verified the protective impact of existing bulk tank testing strategies that are currently being rolled out nationwide. And I would like to emphasize that new information is becoming available every day. And as we receive new data and new ideas, the model and its predictions are subject to change. In conclusion, the risk of contracting H5N1 from consumption of pasteurized milk is extremely low. The risk for raw milk is higher, but additional interventions such as bulk tank PCR testing can reduce this risk. We are investigating additional future directions, especially using some raw milk cheese data to apply to our models. The paper itself is available as a preprint, and this QR code will take you there. Thank you all for your attention, in addition to my lab mates, our collaborators, and the funding sources listed here. Thank you.
Okay, great. Thank you both for the presentations. At this point, we're going to stop the recording and move over to Q&A.